Welcome to Bedtime Astronomy. Explore the wonders of the cosmos with our soothing Bedtime Astronomy podcast. Each episode offers a gentle journey through the stars, planets, and beyond, perfect for unwinding after a long day. Let's travel through the mysteries of the universe as you drift off into a peaceful slumber under the night sky.
Imagine just walking outside tonight. You step away from the streetlights, you look up at the night sky, and you just let your eyes adjust to the darkness.
Yeah, you get that incredible view of the void.
Right, you see the constellations, maybe the faint glow of the Milky Way, just the silent, freezing void of space. But now I want you to imagine realizing that the exact molecular instructions for your eyes to even perceive that light.
Oh, this is the part that just messes with your head.
It really does. The instructions for your heartbeat, the very thoughts forming in your brain right now, they might not have actually originated here on Earth, like not in some warm, muddy puddle.
Which is what we've always been taught, right?
Exactly. I mean, imagine if you found the exact alphabet of your native language, the literal letters you use to communicate, just carved into a pristine rock floating in the absolute zero vacuum of deep space.
It's just wild to think about.
Because, we usually talk about biological origins with this built-in expectation that Earth did all the heavy lifting. We picture our young planet as this magical crucible that just invented life out of thin air. But, what if it didn't?
Well, it completely shatters our traditional Earth-centric view of biology. It forces us to really confront the reality that the alphabet of life was written, or, at least being heavily drafted, in the cosmos, long before our planet was even a hospitable environment capable of reading it.
And we are talking about news that is breaking right now. Today, March 22, 2026. This isn't like theoretical astrophysics or a thought experiment.
Yeah, no, this is hard data.
Right. A groundbreaking study led by Toshiki Koga and his team has just been published in Nature Astronomy. And it drops an absolute paradigm-shifting revelation on the scientific community.
It really does.
They looked at a physical sample brought back from a near-Earth asteroid called Ryugu, and they have confirmed the presence of all 5 fundamental nucleobases, like the entire set.
And the staggering significance here really lies in finding that complete set in a pristine, uncontaminated environment. Because we aren't just looking at a random, chaotic assortment of carbon molecules that might, if you squint, look a little bit biological.
Right, it's not just space sludge.
Exactly. We are looking at the highly specific functional machinery of life. And it's just sitting there on a carbonaceous rock that has been orbiting in space, largely unchanged, for 4.5 billion years.
Okay, let's unpack this.
Okay.
Because before we get into the wild story of how humanity actually managed to fly to a space rock, scoop up dirt, and bring it back, we need to understand the biology.
Yeah, the foundational stuff.
Right. Before we talk about how they managed to find microscopic molecules inside dense rock, we really need to understand what exactly we're looking at. So when we use this term nucleobases, what is the actual physical reality of that? Like if I were to zoom in 1 trillion times, what would I see?
What's fascinating here is that to understand biology, you really have to strip it right down to the chemistry. So nucleobases are essentially organic molecules that contain nitrogen.
Okay, nitrogen.
Right. They are structurally built around carbon rings, but they have nitrogen atoms incorporated directly into those rings, and they form the literal functional letters of genetic information in DNA and RNA.
Right, the famous A, C, T, G, and U.
Yeah.
We learned about them in high school biology. Adenine, cytosine, thymine, guanine, and uracil.
Exactly. And those 5 molecules, they actually belong to 2 distinct chemical families based on their physical shape.
Oh, I didn't realize they had different shapes.
Yeah, they do. So first you have the purines. These are the larger, kind of bulkier molecules because they consist of a double ring structure. If you picture it, they look like 2 hexagons sort of fused together.
Okay, purines are the double rings. Got it.
Right. And that family includes adenine and guanine. Then on the other side, you have the pyrimidines. These are the smaller, more compact molecules made of just a single ring. And that includes cytosine, thymine, and uracil.
So if I'm picturing this, it's almost like a highly specific set of interlocking puzzle pieces. Yeah. But these nucleobases don't just act alone to create life, right? Like a pile of letters doesn't automatically arrange itself into a Shakespearean sonnet just by sitting there.
No, not at all.
They have a very specific recipe they follow.
They absolutely do. In the actual structure of DNA or RNA, A nucleobase doesn't just float freely. It bonds with a sugar molecule like, ribose or deoxyribose, and a phosphate group.
Okay.
And when those three components lock together, the base, the sugar, and the phosphate, you have created what we call a nucleotide.
I want to make sure I'm visualizing this perfectly for everyone. So if we think of genetic material as a book, the nucleobases are the letters of the alphabet, and the sugars and phosphates are essentially the paper and the binding of the book. They hold the letters in a very specific, rigid sequence so they can actually be read.
That is a brilliant analogy, actually.
And when you string millions and billions of those letters together on that paper binding, the resulting epic novel is, well, a living organism.
It is a highly accurate way to visualize the architecture. The sugar-phosphate backbone, it provides the rigid structural rails of the DNA double helix. It's the sturdy frame. Meanwhile, those nucleobases, the letters, they reach across the middle of the helix to pair up with one another.
Like the rungs of a ladder.
Exactly. And because of their physical shapes, the bulky double-ring purines and the compact single-ring pyrimidines, they only fit together in very specific ways.
Right, so they can't just mash together randomly.
No, adenine always pairs with thymine. Cytosine always pairs with guanine. That's what forms those stable rungs of the ladder.
Which is why having all five is so incredibly critical. I mean, it's the difference between having a random pile of ink and actually having a readable blueprint for a skyscraper.
Absolutely. Without these exact 5 molecules, the genetic code that allows an organism to grow, to replicate its cells, to pass on traits to its offspring, and ultimately to evolve over millions of years, it simply would not exist.
Wow.
Life as we know it is entirely dependent on information storage and retrieval. I mean, DNA and RNA are essentially the hard drives of biology, and these 5 nucleobases are the fundamental binary code.
So finding the complete alphabet just existing naturally on an asteroid implies that the universe possesses this fundamental inherent chemistry that leans toward the creation of biological information. The universe is actively forging the letters of life out there in the freezing cold dark.
It forces us to completely reevaluate the chemical potential of the cosmos. I mean, it tells us the ingredients for biology are not some freak localized accident that only happened on a warm Earth.
Which transitions us perfectly from the microscopic biology happening inside our cells to a massive macro-level space mission. Because, we know what these molecular letters are now, and we know why they are basically the holy grail of biology. But how did we actually get our hands on them?
Yeah, that's a whole other incredible story.
Right. We didn't just point a really strong telescope at Ryugu and see little molecular letters waving back at us. This required an actual physical cosmic heist.
A profoundly difficult, highly calculated cosmic heist, yes. This was the Hayabusa 2 mission, which was conducted by JASA, the Japan Aerospace Exploration Agency.
Okay, so Japan led this.
Right. They launched this spacecraft back in 2014, and it spent nearly 4 years navigating millions of miles through deep space just to intercept this one specific asteroid, Ryugu.
And Ryugu isn't just a smooth, friendly little moon, right?
Mm-hmm.
From the images I've seen, it looks terrifying.
Yeah.
It's a pitch black, shaped kind of like a spinning diamond or a top, and it's just covered in jagged boulders.
It is essentially a flying rubble pile. It's about 1 kilometer wide, composed of rocks and dust that are only loosely held together by its own very weak gravity.
Wow.
So when Hayabusa finally arrived in 2018, the team realized the surface was far too rugged for a traditional smooth landing. They had to spend months painstakingly mapping the surface just to find a safe, tiny patch of gravel.
And the collection method itself was basically a drive-by shooting. The spacecraft didn't land and deploy a little rover with a shovel. It hovered just above the surface, descended rapidly, and the moment its sampling horn touched the asteroid, it literally fired a tantalum bullet into the rock.
Yes, exactly. Firing that projectile kicked up this cloud of subsurface dust and debris, and that material was then funneled up the horn and caught inside a sterilized collection chamber.
Like a vacuum cleaner in space.
Pretty much. And then the spacecraft immediately fired its thrusters to back away, sealed the chamber, and began the long journey back to Earth. It eventually dropped the sample return capsule into the Australian outback in late 2020.
Okay, but here is where I really have to pause, because when I first saw the numbers on this mission, I almost didn't believe it. I want you, the listener, to really picture this. They spent hundreds of millions of dollars, flew a spacecraft for 6 years, shot an asteroid with a bullet, and brought back a grand total of 5.4 grams of material.
Yeah, it doesn't sound like much.
That's roughly the weight of a nickel. I have to ask the obvious question. How can we possibly learn anything about the origins of life, or the 4.5 billion year history of the solar system, from a literal teaspoon of space dirt.
Well, when you picture a space mission bringing back samples, the cultural image we all share is like the Apollo astronauts lugging massive, heavy crates of moon rocks out of the capsule.
Right, exactly. Big gray rocks.
So 5.4 grams sounds like a failure to a lot of people. But in the realm of modern organic chemistry and cosmochemistry, 5.4 grams is a treasure trove. It is an almost unimaginable abundance of material to work with.
How is that even possible? Is it just because our laboratory instruments are that sensitive now?
Correct. That's a huge part of it, yes. Analytical chemistry has advanced to a point where we basically no longer need boulders to read the history of the solar system.
Oh, wow.
We have these highly advanced mass spectrometers and liquid chromatography techniques today that can detect organic molecules at the parts per billion or even parts per trillion level.
Parts per trillion. That's insane.
It really is. A single grain of sand from that asteroid provides enough material to run dozens of exhaustive chemical analyses.
So the mass of the sample isn't really the limiting factor at all.
Not at all. The true value of that 5.4 grams isn't its weight, it's its history. Asteroids like Ryugu are classified as carbonaceous chondrites.
Carbonaceous chondrites.
Right. They are essentially the pristine, leftover construction debris from the very formation of the solar system.
They're the scraps left over on the workshop floor after the sun and the planets were built.
Exactly. And because Ryugu is so incredibly small compared to a planet, it never underwent the extreme geological heating, the melting, and the tectonic recycling that bodies like Earth or Mars did.
Oh, because Earth is geologically active.
Right. When Earth formed, it became a massive molten ball of magma. Any fragile organic molecules present at its birth were completely incinerated.
It burned to a crisp.
Yep. Ryugu, on the other hand, has essentially acted as a deep freeze time capsule. It's preserved the chemical environment of the early solar nebula, largely unchanged, for about 4.5 billion years.
Okay, but if you're listening to this and wondering why scientists care so much about flying to an asteroid when meteorites hit the Earth all the time, I had the exact same thought. I mean, people find meteorites in Antarctica, in the Sahara Desert, literally in their backyards.
I do, constantly.
Right. The universe is throwing free samples at our planet every single day. So why spend a decade and $1 billion to go out to Ryugu?
That touches on what is arguably the most frustrating problem in the entire history of astrobiology: contamination. The crucial distinction between a meteorite you pick up off the ground and the sample brought back from Ryugu comes down to that word, pristine.
Because the moment a meteorite enters Earth's atmosphere, it's compromised.
It is violently compromised. First, it gets blasted with thousands of degrees of heat as it plummets through the atmosphere, and then the very second it touches the ground, it is instantly exposed to Earth's rich, aggressive, pervasive biosphere.
Just totally swarmed.
Exactly. Bacteria immediately colonize it. Fungi spores land on it. Amino acids from the surrounding soil seep into its porous rock structure with the very first rain.
Oh, wow. So if you pick up a carbon-rich rock from the desert, bring it to a lab, and find nucleobases, the letters of DNA, hidden inside it, you are instantly hit with this lingering, annoying doubt. Like, did this alphabet come from space, or did a microscopic soil might just sneeze on it yesterday?
Exactly. And that doubt has plagued cosmochemists for over a century. You are constantly forced into a position of trying to prove a negative.
Which is impossible.
Right. Trying to mathematically and chemically prove that the biological molecules you found inside the meteorite aren't from Earth. And skeptics will always find a reason to doubt it.
I mean, fair enough.
But the Hayabusa 2 samples bypassed the biosphere entirely. They were collected in the pure vacuum of space, sealed in a pristine container, parachuted into the desert, and brought directly into sterilized laboratories before they ever even touched Earth's air.
Wow.
There is no asterisk next to the data. It is a pure, untainted look at the solar system's ancient chemistry.
Which takes us right to the door of the laboratory, because even having secured this pristine 5.4 grams, this precious nickel's worth of the universe, how do you actually study it? I mean, if these microscopic letters of life are hidden inside solid, dense rock, you can't just throw the rock under a microscope and start looking for A, C, T, G, and U.
Definitely not.
How do scientists actually get the molecules out without completely destroying the sample in the process?
Well, it requires a level of paranoia regarding cleanliness that literally borders on the obsessive. We are talking about ultra-clean lab conditions. These facilities are orders of magnitude cleaner than a hospital operating theater or like a silicon microchip manufacturing plant.
Because if a scientist breathes a little too heavily, or if a single stray speck of Earth dust from the ventilation system enters the lab, or if there's a microscopic flake of dandruff.
It's game over.
Yeah. The entire 4.5 billion-year-old narrative is immediately compromised. You've just ruined the most expensive dirt in human history.
To prevent that, the scientists never actually touch the asteroid grains, and the grains never touch the air we breathe. The extraction process operates entirely inside these specialized, sealed glove boxes.
Like those boxes where you stick your arms into the big rubber gloves.
Exactly those. But these boxes are constantly purged and filled with inert, purified nitrogen gas.
Wait, why nitrogen specifically?
Because the oxygen and moisture in our normal air are highly reactive. If Earth's atmosphere touches those 4.5 billion-year-old grains, it can instantly trigger oxidation reactions, fundamentally altering the ancient organic molecules before we even get a chance to measure them.
Oh, so the air itself would rust the molecules, basically.
In a sense, yes. So the environment must be completely inert.
Okay, so you have the rock sealed in a nitrogen box, but you still have to get the molecules out of the rock. It's almost like brewing the most delicate, high-stakes cup of tea in the entire universe. You literally have to steep the rock right.
The brewing analogy is actually highly accurate. The extraction methodology relies on liquid solvents to pull the organic molecules out of the solid rock matrix. In the case of Toshiki Koga's research, they utilize two sequential steps: hot water, followed by hydrochloric acid.
Okay, water makes sense. I mean, you make tea with hot water. But hydrochloric acid, that sounds incredibly violent. Why would you use both?
This raises a really important question about the physical nature of these molecules within the asteroid. So the hot water extraction is relatively gentle. It penetrates the rock and pulls out the free-floating, easily soluble organic compounds that are just resting loosely within the pores of the asteroid.
The easy stuff.
Right. But cosmochemists know that a massive portion of these complex organics aren't just resting loosely. They are deeply bound up within the solid, insoluble mineral matrix of the asteroid. They are literally trapped inside microscopic cages of silicate rock.
And hot water isn't strong enough to break a rock cage.
Exactly. So to access that hidden inventory of molecules, you have to introduce the acid. The hydrochloric acid effectively dissolves the surrounding silicate minerals. It melts away the rocky cages, liberating those trapped organic molecules into the liquid solution without completely destroying the organics themselves.
Wait, hold on. How does boiling a nucleobase in hydrochloric acid not destroy it?
Yeah.
I mean, if you dropped a modern strand of DNA into acid, it would just disintegrate.
And that illustrates just how incredibly robust these individual precursor molecules must be. The nucleobases themselves, the individual letters, are highly stable ring structures.
They're just that tough.
They really are. They can survive the intense radiation and freezing vacuum of deep space for billions of years, the violent extraction from an asteroid via tantalum bullet, the journey to Earth, and then being soaked in acid in a laboratory. And they still maintain their chemical integrity.
That is wild. So after you've made this incredibly complex, highly acidic space tea, how do you actually read the letters floating in it?
Following that brutal extraction, the resulting chemical soup is highly chaotic. It contains thousands of different types of molecules, dissolved minerals, and just a lot of background noise. So it has to be painstakingly purified using liquid chromatography.
Which does what exactly?
It separates the molecules based on their size and chemical properties. Once you have isolated the specific fraction of the liquid that should contain nucleobases, you feed it into a high-resolution mass spectrometer.
Okay, for the listener who, like me, hasn't taken a chemistry class in a decade, how does a mass spectrometer actually find an invisible molecule?
It's an incredibly elegant piece of machinery. It essentially vaporizes the liquid sample, gives the individual molecules an electric charge, and then shoots them down a vacuum tube through a powerful magnetic field.
Okay, shooting charged molecules through a magnet.
Right, and depending on how heavy a molecule is, its mass, and the charge it carries, the magnetic field will bend its path by a very specific, mathematically predictable amount. Lighter molecules bend more, heavier ones bend less.
Oh, I see.
By measuring exactly where they hit a detector at the end of the tube, scientists can determine the precise molecular weight of the substance. And since we know the exact molecular weight of adenine or cytosine or uracil, we can look for that specific unique signature in the data.
Here's where it gets really interesting, though, because this whole extraction and detection process wasn't just an overnight eureka moment where they turned on the mass spectrometer and all 5 letters of the genetic alphabet just popped up on a screen simultaneously.
No, it was a long haul.
There is a timeline to this discovery that I find fascinating. Back in 2023, an international team reported finding just one of the nucleobases in the Ryugu samples. They found uracil, and it made massive headlines.
It was huge news.
But it's now 2026, and Toshiki Koga's team has finally confirmed all 5 are present. Why did it take 3 full years to go from finding uracil to finding the rest of the alphabet? Were the other molecules just hiding deeper in the rock?
Well, it beautifully illustrates the iterative, highly cautious nature of scientific discovery. Finding uracil first actually makes perfect sense from a purely chemical perspective.
Really? Why is that?
Remember earlier when we divided the nucleobases into 2 families, the purines and the pyrimidines?
Yeah, the double rings and the single rings.
Exactly. Uracil is a pyrimidine. It's a single ring structure. It's a smaller, somewhat simpler molecule. Cosmochemical models show that in the cold, harsh environment of space, the prebiotic chemical reactions required to build a simple single ring can occur much more readily than the complex reactions required to build a bulky double ring purine like adenine.
Okay, that makes sense.
Consequently, there is simply more uracil present in the sample to begin with. The signal is louder.
So uracil is basically the low-hanging fruit of the molecular alphabet.
In a sense, yes. Detecting the others, particularly the larger purines, requires pushing the analytical techniques to their absolute breaking point. The concentration of these molecules is astonishingly low.
How low are we talking?
We're talking about detecting them at a few parts per billion. To give you a sense of scale, that is like trying to find a single specific grain of sugar dissolved into an Olympic-sized swimming pool.
Wow.
The background noise from the thousands of other random non-biological carbon molecules in the asteroid can very easily drown out the incredibly faint signal of guanine or cytosine.
So the 2023 discovery of uracil was basically a proof of concept.
Precisely. It told the global teams, look, the underlying prebiotic chemistry is here. The mechanisms to make nucleobases exist on Ryugu. Now we need to go back to the drawing board, refine our purification methods, adjust our solvent ratios, and tune the mass spectrometers to look specifically for the much fainter signatures of the other 4.
And it took 3 years to do that.
It took 3 years of relentless laboratory refinement to achieve the sensitivity required to reveal the complete set.
And one of the most vital details of this 2026 study is where they found them. They didn't just find all 5 in one lucky little speck of dust. They analyzed 2 completely separate samples taken from different locations on the Ryugu asteroid, and they found all 5 nucleobases in roughly similar amounts in both.
And that detail is the linchpin of the entire study.
Yeah.
If you only analyze one tiny grain from an asteroid and find these complex biological precursors, the scientific community will immediately push back.
They'll call it a fluke.
Exactly, they will ask. Was that single grain special? Did it undergo some freak, highly localized chemical reaction, maybe a microscopic comet impact, or an unusual radiation spike that just isn't representative of the rest of the asteroid?
It's like taking a blind bite of a cake.
Yeah.
If you take one bite and get a huge chunk of chocolate, you don't know if the whole cake is chocolate, or if you just happen to hit the one single chocolate chip the baker accidentally dropped into a vanilla batter.
That's exactly it.
But by taking a sample from the north pole of the asteroid and another sample from the equator and finding the exact same chemical alphabet in roughly similar concentrations.
You mathematically prove the cake is chocolate through and through. Finding identical molecular profiles in 2 disparate locations completely eliminates the possibility of a localized fluke or a detection error.
It makes it a rule, not an exception.
Right. It proves that these organic molecules are uniformly distributed within the matrix of the asteroid. And that means the chemical processes that created them weren't a rare, localized accident. They were a ubiquitous, widespread phenomenon happening throughout the entire parent body of that asteroid billions of years ago.
Which opens up a massive, profound question. Because Ryugu isn't the only rock we've studied. It's part of a much larger cosmic family. So how does Ryugu's chemical profile stack up against the rest of the solar system?
That's the logical next step.
Because to truly understand this discovery, we have to look at the tale of 4 major space rocks that cosmochemists have just been obsessed with. We have Ryugu, we have the recent samples from asteroid Bennu, and we have the historical meteorites that crashed into Earth decades or even centuries ago.
And comparing Ryugu to the historical meteorite record is what really grounds this modern discovery and gives it massive historical weight. Because long before we had spacecraft capable of sampling asteroids, we had meteorites. For decades, the absolute gold standard for cosmochemistry was the Murchison meteorite.
That's the one that fell in Australia, right?
Yes. In 1969, It fell near a small cattle town called Murchison in Victoria. It was a massive event. Over 100 kilograms of rock shattered over the countryside.
That's huge.
And it fell just months after the Apollo 11 moon landing, so laboratories around the world were already primed and equipped to analyze extraterrestrial rocks. When they analyzed Murchison, they were stunned to find a rich variety of organic molecules, including amino acids and nucleobases.
But there was an even older one, right? A legendary meteorite.
Oh, the Argyll meteorite. This is perhaps the most famous carbonaceous chondrite in history. It fell in southern France in May of 1864.
1864.
Think about the historical context there. The American Civil War was raging. Louis Pasteur was conducting his famous experiments proving germ theory. And suddenly, this massive charcoal-like rock explodes over a French farming village, scattering pieces across the peat bogs and fields.
And didn't people immediately think it had life in it?
They did. When scientists in the 1800s first analyzed it, they found it was packed with complex carbon compounds. Some even claimed to see microscopic fossils of bacteria inside the rock.
No way.
Yeah, it kicked off a 160-year debate. Because Argyll fell into the muddy French countryside, and Murchison fell into the Australian dirt. For a century and a half, scientists have been studying these two meteorites, always hounded by the contamination problem.
Like we talked about earlier.
Exactly. Are we studying ancient space chemistry, or are we just studying French soil bacteria that crawled into the rock in 1864? Are these nucleobases from the early solar system, or from an Australian cow pasture?
But now you have the Ryugu samples from 2020. And to add another layer of confirmation, in 2023, NASA's OSIRIS-REx mission returned pristine samples from another carbon-rich asteroid called Bennu, which were fully analyzed by 2025. And those Bennu samples also yielded all 5 nucleobases in a pristine, uncontaminated state.
And that provides a beautiful, deeply satisfying, full-circle moment for the scientific community. The pristine space samples from Ryugu and Bennu serve to retrospectively validate the findings from Murchison and Argyll.
It proves they were right all along.
By matching the chemical profiles, we now know definitively that those historical meteorites were indeed carrying authentic extraterrestrial nucleobases all along. The French scientists in 1864 really were holding the chemical precursors of life in their hands. They just couldn't definitively prove it.
But here is where the plot of our cosmic story really thickens. Because while they all contain the molecular alphabet, they aren't exact copies of each other. The chemistry isn't just a simple carbon copy from rock to rock.
No, there are significant differences.
Kojiki Koga's research highlights that Murchison is heavily enriched in purines, those bulky double-ring structures like adenine. But Bennu and Argyll contain a much higher concentration of pyrimidines, the compact single-ring structures.
And that variance is one of the most exciting aspects of the data. It tells us the solar system is not a monolith.
It's almost like looking at different regional cuisines on Earth. They all start with the same basic ingredients: flour, water, salt, yeast. But the local environment, the local heat, the humidity, the local culture, it fundamentally changes the final dish.
Right.
One region in Italy makes a delicate pasta. A region in California makes a dense sourdough bread. They're using the exact same building blocks, but the outcome is totally different.
The regional cuisine analogy is perfect. The basic, fundamental building blocks of carbon, nitrogen, hydrogen, and oxygen were ubiquitous in the early solar nebula, the vast cloud of dust and gas that formed our sun and planets. But the local environment inside the specific asteroid dictates the final chemical dish.
So if these rocks are all essentially born from the same early solar system dust cloud, why does Murchison serve up a dish heavy on purines, while Bennu and Argyll serve up pyrimidines?
Wow.
What is the master chef in this scenario? What is the specific ingredient actually causing that chemical divergence?
Well, the current consensus among cosmochemists points heavily to a specific, highly influential molecule acting as the master chef of this prebiotic chemistry: ammonia.
Wait, ammonia, like the chemical we use in cleaning supplies.
Exactly that. Ammonia, chemically known as NH3, consists of one nitrogen atom bound to 3 hydrogen atoms, and it is incredibly abundant in the colder regions of the solar system. The balance between purines and pyrimidines inside these asteroids is thought to be deeply dictated by the concentration of ammonia present when the asteroid was chemically active.
How does a molecule like ammonia actually dictate which nucleobase forms? Does it physically alter the environment inside the rock?
It does something extraordinary. It acts as a cosmic antifreeze.
An antifreeze.
Yeah. Deep inside these asteroids, shortly after the solar system formed, there were pockets of ice. As radioactive elements inside the rock decayed, they produced heat, melting the ice. If you have pure water inside a freezing asteroid, it will quickly freeze solid again. But if that water is heavily mixed with ammonia, the freezing point drops drastically. Ammonia allows liquid water to persist inside a freezing space rock for millions of years.
And liquid water is the absolute prerequisite for context chemistry.
Right.
You can't run a chemical reaction in solid ice. The molecules have to be able to swim around and bump into each other.
Precisely. Furthermore, ammonia doesn't just keep the water liquid, it actively participates in the chemistry. It acts as a primary nitrogen donor.
Oh, because nucleobases need nitrogen.
Right. Remember, nucleobases are carbon rings with nitrogen atoms sprinkled in. The more ammonia you have in the liquid water, the more freely available nitrogen you have to donate to the growing carbon structures.
So it's feeding the reaction.
Exactly. So if an asteroid forms in a region of the solar system with a very high concentration of ammonia, that ammonia will catalyze the complex reactions required to build the bulky purines. If an asteroid forms with less ammonia, the chemistry leans heavily toward the simpler pyrimidines.
So what does this all mean for us? If ammonia acts like the master chef, deciding the exact molecular menu based on local conditions, and we know that different carbon-rich asteroids throughout the solar system contain entirely diverse prebiotic chemistry dependent on their specific history.
Yeah.
Does that mean life could have looked fundamentally different if Earth had been seeded by a different asteroid?
If we connect this to the bigger picture, that is the exact profound implication of these chemical differences. The early solar system wasn't just a uniform, homogeneous soup of matter. It had distinct chemical zones, almost like weather patterns in the early solar nebulae.
Chemical weather, I love that.
Yeah, an asteroid forming further out past the orbit of Jupiter might have accumulated vastly more frozen ammonia, fundamentally changing its internal chemistry compared to an asteroid forming closer to the young sun.
So the specific chemical weather inside the asteroid dictates its biological payload.
Yes. The unique chemical history of a parent body dictates its prebiotic payload. It proves the chemical building blocks are highly sensitive to their environment. And that has massive implications for the origins of life on Earth.
Because if the early Earth had been bombarded exclusively by asteroids that were severely depleted in the precursors for, say, pyrimidines, the primordial soup on our planet would have been chemically skewed.
Heavily skewed.
The emergence of DNA as we currently know it, which requires a perfect, equal pairing of purines and pyrimidines, might have been drastically delayed.
Or life might have had to find a completely different method of information storage based on a different molecular architecture entirely. The chemistry of life on Earth is a direct reflection of the specific population of asteroids that happen to crash into it.
That is just mind-bending. The idea that the very physical structure of our DNA, the exact shapes of the genetic ladder that makes you and me human, was highly contingent on the specific chemical weather happening inside a rock floating in the void of space billions of years ago.
It really puts things in perspective.
Which brings us to the final and perhaps most philosophical piece of this massive puzzle. The delivery system to early Earth. We've analyzed the chemistry, we've broken down the extraction methods, we've explored the asteroids. Now we have to bring it home. How do these findings actually reshape the story of our own origins?
Well, traditionally, when we learn about the origin of life in school, we are taught the primordial soup model.
Right, the lightning hitting the puddle.
Exactly. The idea is that early Earth was a warm, wet environment with a chaotic atmosphere. Lightning strikes and volcanic heat provided energy, and over millions of years, simple gases slowly combined to form complex molecules, eventually resulting in the first living cell.
Right.
It's a very localized, closed system view. Earth is the laboratory, Earth provides the raw ingredients, and Earth cooks the meal entirely on its own.
But the discovery of all 5 nucleobases on Ryugu and Bennu in the meteorites, it violently disrupts that closed system model. It suggests that the molecular ingredients of life were already forming in space billions of years ago, entirely independent of Earth.
Yes. It introduces a scientific framework closely related to a concept called soft panspermia.
Panspermia being the theory that life travels across the universe.
Correct. Now, hard panspermia suggests that fully formed, living, reproducing bacterial cells originated in space and were carried to Earth on meteors.
Like actual alien microbes.
Right. But there is very little evidence to support that extreme view, as the radiation of deep space would likely kill a living cell over those vast timescales. But soft panspermia strongly implies that while life itself didn't come from space, the highly complex, absolutely necessary ingredients for life were manufactured in the cosmos and delivered to early Earth.
But wait, how does a delicate molecule like a nucleobase survive the delivery? We talked earlier about how meteorites are blasted with thousands of degrees of heat when they enter the atmosphere. We see them as shooting stars. Wouldn't the heat of atmospheric entry, followed by a violent crater-forming impact, just instantly vaporize these fragile organic molecules?
It seems highly counterintuitive, I know, but physics provides a fascinating mechanism called ablative shielding.
Ablative shielding.
When a large meteor enters the atmosphere, the friction generates immense heat. But that heat primarily vaporizes just the very outer layer of the rock. The rock is ablating, burning away, but rock is actually a terrible conductor of heat.
Oh, like how you can hold a hot cup of coffee if it has a thick ceramic mug.
Exactly. The outer few centimeters of the meteor reach thousands of degrees, but the interior of the rock, deep inside, remains effectively at the freezing temperature of deep space, all the way down to the moment of impact.
That's incredible.
It's almost like a cosmic baked Alaska, scorched on the outside, freezing cold on the inside. So the organic payload hidden deep within the pores of the rock survives the journey perfectly intact.
And once it impacts the early Earth, the rock eventually breaks down, interacts with water, and releases that pristine alphabet into the primordial oceans.
And this delivery wasn't just a rare occasional event. About 4 to 3.8 billion years ago, the solar system went through a period known as the Late Heavy Bombardment.
The Late Heavy Bombardment.
Right. Planetary orbits were shifting, gravitational pulls were chaotic, and millions of asteroids and comets were thrown into the inner solar system, pummeling the early Earth incessantly.
It changes everything about how you view the sky. If you're listening to this and you go outside during the Perseid or the Leonids meteor shower, you look up and see those beautiful streaks of light burning up in the atmosphere. You aren't just looking at pretty space debris.
No, you're not.
You might literally be looking at the exact same delivery mechanism that brought the genetic code to a barren, hostile early Earth.
During the Late Heavy Bombardment, it was quite literally raining organics. The story of life on our planet is deeply, inextricably connected to the chemistry of these ancient asteroids. Life didn't necessarily have to invent its own alphabet from scratch, struggling through trial and error in a primordial soup on a young, unstable Earth.
The alphabet was already written in the stars.
It was synthesized in the warm, wet interiors of primitive asteroids, catalyzed by ammonia, preserved in a deep freeze, and delivered via a massive cosmic bombardment. It redefines our planet not as a lonely, isolated miracle that managed to do all the work itself, but as the incredibly fortunate beneficiary of a much larger, active, cosmic, chemical story.
It is just a breathtaking journey to wrap your head around. I mean, we started by simply defining the A's, C's, T's, G's, and U's, the fundamental letters of our genetic code, the specific shapes of purines and pyrimidines.
Yeah, the basic building blocks.
And then we traced the astonishing human ingenuity required to extract those exact microscopic letters from just 5.4 grams of a 4.5 billion-year-old pristine rock, flying millions of miles, shooting an asteroid with a tantalum bullet, utilizing nitrogen glove boxes, hot water, and acid just to reveal the invisible chemistry inside.
And through that painstaking decades-long process of comparing Ryugu to historical meteorites like Murchison and Orgai, we realized that our biological origins, the very essence of what makes us living, replicating organisms, are intrinsically linked to the solar system's ancient chemical weather.
The balance of ammonia acting as an antifreeze and a master chef.
The temperature gradients of the early nebula, the unique cosmic journeys of the rocks that seeded our oceans.
It makes you realize how utterly connected we are to the void above us. We aren't just on Earth. We are made of the solar system. So I want to leave you, the listener, with a lingering thought to mull over today as you go about your life. If carbon-rich asteroids, scattered by the millions across our solar system, are all independently acting as these tiny, perfect cosmic chemical laboratories, brewing up the exact molecular alphabet required for life in the absolute dark, what are the odds that Earth was the only planet in the universe to actually open the book and start reading?